Okay, so it has been a while since I've done one of these, but I'd figure I enjoy making these videos so much that I'd revisit going over some amino acids. So let's get started. Let's just go from the top, and let's start with the non whoops, and let's start with the non-polar and hydrophobic amino acids. So the general structure of an amino acid is, of course, we have the amino group or the amine group. Typically, you'll find this is protonated at physiological pH, which is around 7.2. So this would be positively charged, and of course we have the carboxylic acid portion, which is usually negatively charged. So we could write it like that, not negatively charged at physiological pH. So anyway, let's start with alanine. So this is alanine, right? So alanine is just kind of one of those special cases where there isn't really an easy way to remember it. It's just that this is one of the simplest amino acids, A for alanine, right? There's really no easy way to memorize this, unfortunately, but it is one of the non-polars. So yeah, moving on to valine. Valine, there is an easy way to memorize this, and of course here's the reference of amino acids over here. For valine, just imagine the name, valine. Right, so V for valine, that's a one-letter code, and it makes a little V shape, right? So let's highlight that in red. So it makes a little V shape. So V for valine, and that is nonpolar, right? Similarly, there's an amino acid that looks like valine but is slightly different, and that of course is threonine. So threonine is down here in the polar amino acids. So threonine. So just imagine, just write out the name, threonine, and look here, threonine. There's an oxygen here. So think threonine, threonine. That has an oxygen in it, but it has a similar structure to that of valine as well. So of course, the difference between this one and valine is that this one is polar, so therefore it's going to be attracted to water, versus valine, which is going to be attracted to fat. So you will likely see valine on the inside of you will likely see valine on the inside of or embedded into a cell membrane, because the inside of a cell membrane is fatty, or you will see valine folded inward in a protein because water repels it, versus threonine, which will of course be attracted to water. And as in addition to that, threonine is also able to be phosphorylated. And of course, when something is phosphorylated, it could have a number of different functions. Usually it's for cell signaling, or usually it's for just making sure a protein is more... usually when there's a phosphate, it means it's a better leaving group, so it's more reactive. It can be used in enzymes, all sorts of different reasons why this could be phosphorylated. But just know that if it has an OH group in the residue, it is likely able to be phosphorylated. Which kind of brings me to the next amino acid, which of course is tyrosine. I'm not really going in any particular order here. I'm just kind of going coming as I go. And of course, with tyrosine, you could use the same mnemonic. So tyrosine, that has an OH in it, and similarly, tyrosine can be. Sorry, one sec. Similarly, tyrosine can be phosphorylated, and for the same reasons, the phosphorylation can. Similarly, the phosphorylation is important for, cell signaling and all of that. So moving on, let's continue with the polar groups. So of course we have up next serine. So serine, kind of like alanine, is one of those amino acids that are pretty simple in their structure, and there's no real easy way to memorize them. Just remember, like, alanine is short, serine is short, cysteine is short. They only have just the one carbon with something attached to it, right? Or in the case of alanine, just the one carbon. So this would be one of those examples. So serine would have the oxygen attached to it, and if you peel back a couple layers and you actually put up a thio group, you do get cysteine, right? And that's next on the list, right? So cysteine, of course, has the very important function of being able to make disulfide bridges. This is very important for the tertiary level of binding, of folding in a protein. And it does have negative side effects like aggregating in your kidneys as kidney stones. If you have too much cysteine, then it will aggregate. So yeah, this is the reduced form. They also call it sulfhydryl, which sulfur hydrogen makes sense. Sulfhydryl. You can also you also hear thio or mercapto when you see this group. Personally, I like thio because you will see the name thio in nearly every sulfur-containing compound, which you will see later. But anyway, this sulfhydryl group, this is the reduced form, and once it binds to another sulfur, it becomes oxidized. So this is the reduced form, and this is the oxidized form. Okay, so moving on. Actually, I did say that the word thio does come up sometimes, and here to prove that is methionine, right? So methionine is over here, right, in the non-polar region. So despite the fact that sulfur is a little more electronegative than carbon, it doesn't mean it's going to be a polar molecule. So you can see this, it's usually going to be nonpolar. And methionine has an important role in usually being one of the first amino acids that is translated in the ribosome, right? So the methionine, like if you remember AUG, the code in RNA, that codes for methionine. Usually methionine will be first. And methionine is, of course, nonpolar, and like I said, methionine, the thio indicates that it's a sulfur-containing compound. Okay, moving on. Just one moment. I'm using Microsoft Paint right now, and unfortunately, Microsoft decided to actually ruin it, so you have to bear with me. Moving on, I went over tyrosine. I already went over tyrosine, but let's also go over a similarly shaped amino acid, which of course is phenylalanine. So when you see a group like this, so this is a benzene group, of course, and if you take a look at alanine, right? Alanine is a pretty simple structure. So if you attach a benzyl group, or in this case a phenyl group, which is another name for a benzene ring, if you attach a phenyl group, then you get phenylalanine. And of course, despite the fact that it's aromatic, it might have some. With aromatic compounds, there is a chance that it's actually pretty interesting because you do sometimes see another aromatic group. They sometimes interact with the rings. It's really cool, and it could be used in some new drug research. But anyway, for this aromatic compound, generally it'll just be nonpolar. Versus if you add up top here an OH, it becomes tyrosine. But this, regardless, that was phenylalanine. So moving on. So two amino acids that you're not as likely to see on the exam are going to be asparagine and glutamine. So asparagine and glutamine are two amino acids in which they have these amino groups as well. But these amino groups, because this is a amide, it has a lot. This is an amine. This would be an amide. So an amide has the carbonyl group, and with the carbonyl group is attached a nitrogen. This is an amide. So usually with amides, there's less of a chance there will be a protonation here, for the same reason that an aldehyde, has a very low chance of being ionized. In the same way, this also has a low chance of being ionized. So unfortunately, with asparagine and glutamine, and later when we go over glutamic acid and aspartic acid, usually, I mean, unfortunately, there's no easy way to remember it, because the thing about those two is that they're very similar, and there's only one difference. In fact, I'm going to draw both of them just to show you how similar they are, right? So I always forget. I think glutamine has 1, 2, and then so look how similar these are. The only difference is that asparagine has one carbon and one carbon in the bridge, and glutamine has 2. But in the names, at least, you could they indicate their structure a little bit. So glutamine has an amine. Asparagine also has an amine. So at the very least, they somewhat indicate their structure. And in the same way, there's also glutamate and aspartate, or glutamic acid and aspartic acid. They mean the same thing, except that I'll just draw it out now that I'm talking about them. Just one moment, please bear with me. So with aspartic acid and glutamic acid, or glutamate and aspartate, the only, like I said, they're very similar to each other. The only difference is the one carbon, the one extra carbon, which is located here and here. So this is aspartate, and this is glutamate. And earlier this was asparagine, and this was glutamine, right? So if you know a little bit about food and cooking, you might have heard about MSG, right? So MSG, it stands for monosodium glutamate. So that is just this amino acid, just a single amino acid with a sodium ion attached to it. And apparently it gives off this really umami flavor. It tastes really good. In fact, the umami receptor in your tongue is what detects glutamate. So yeah, glutamic acid and aspartic acid, I think the way to remember the function specifically is that, well, for one, they're negatively charged, so they're able to form salt bridges. So of course opposites attract, so once you have a positive charge and a negative charge, they're going to want to bind together. So typically when you have an aspartate and you have something like a histidine, they are able to bind with each other ionically. And ionic bonds can typically be quite strong. So just think, aspartic acid, glutamic acid, those are the ones that are able to be ionized, since they, of course, are acids. And once you have the ionized form of an acid, then you add the suffix "ate" to it. So another example of that would be, like, think of acetic acid. The ionized form of acetic acid, which is vinegar. The ionized form of acetic acid would be acetate. Let me think of another example. The citric acid, the stuff that makes lemons so sour. Citric acid, once ionized, is, of course, citrate. So in the same sense, aspartic acid, aspartate, glutamic acid, glutamate. And these ones are going to be pretty important. And I think, unfortunately, there is no way to just remember. There is no easy way to remember that aspartate has 1 and glutamate has 2. It's just something. Unfortunately, that's just yeah. But the function is that they form salt bridges, and they are negatively charged ions. They can be negatively charged ions once in physiological pH. Anyway, let's redraw our amino acid, since I had to delete the other two. So moving on, let's move on to now that we talked about the acidic group of amino acids. Let's talk about the basic. So the basic and acidic amino acids, they fall under the same group of just being ionically charged. But of course, you can go a little further than that and say one is negatively charged, would indicate that it's acidic, and one is positively charged, which would indicate that it is a basic amino acid. So let's move on to histidine. So of course, histidine sounds a lot like histamine, right? And histamine is the one thing that is the bane of many during allergy season, right? And it could also be the bane of many when eating food that is unfamiliar. That looks about right. I think I'm missing one. I always have a hard time drawing this one. And then there's okay, so 1, 2, 3, 4, 5. Yeah, okay, this is right. Anyway, histidine. In histidine, I believe it's this. I believe it's either this or this that could be ionized. Anyway, let's assume it's this one. So once this, wait, I'm looking at it and it's wrong. I'm sorry, this is gonna bother me. Okay. Okay. I see the problem, I think. Once again, they call it an imidazole ring that histidine has. Like it's a specific like functional group that shows up every now and then. So anyway, and then let's go here. And that's one group, and then this is directly attached to nitrogen, and then we have just like that. Okay, there we go. This is our imidazole ring. And this one has a hydrogen, and this one could be positively charged to also have a hydrogen. So this, right, so histidine has a pKa of around 6.4, which means at physiological pH, it's going to be in the ionized form. So it's going to act like at physiological pH, it's likely going to be positively charged. But because it's so close to physiological pH, it could actually be readily ionized or unionized. It's kind of cool. So histidine, of course, like this is one of the weird looking ones. This is one of the kind of like you could say it's a cyclic amino acid. I'm not sure if I would go that far. It's definitely basic because it does have the ability to be charged. So in this case, right, like the positively charged amino acids are of course going to be able to form salt bridges with something like glutamic acid and aspartic acid. So histidine does have that capability, and histidine also has the unique factor that like once you modify it a little bit, it becomes histamine, right? So histamine, just think of this imidazole ring. Histidine, histamine, imidazole ring. You can make a little rhyme in your head with it, and just also remember that it is positively charged at physiological pH. So moving on from that, we have two kind of unique amino acids. They are also positively charged. Arginine, right? So let's see. 1, 2, 3, 4, 5. And then it has what we call like an imine group. So it has like this double bonded nitrogen into an amine group. So asparagine also can be positively charged. And I think it's here at the imine group that we're able to have another where we'll have another hydrogen. Sorry if my drawing is not up to snuff. It's been a while since I've done this. Anyway, I always like to say arginine. Like arginine's code is the single letter code is R. So arg, walk the plank. That's how I like to think of it. So think of it like a pirate anyway. So arginine does have that unique factor to it also that it is able to, I guess not really unique, but it does have that ability to have a positive charge. So and of course the pKa of this particular structure is going to be around 11 or 12. I can't really remember. So this is almost always going to be ionized in nearly every part of the body. This is an incredibly difficult structure to deionize. So yeah, if you see a body and that part of the body has the unionized form of arginine, something's gone seriously wrong. Anyway, so moving on to lysine. So lysine in a similar way is also quite long and also is able to be positively charged because of the amino group at the end. So okay. Let's redraw this as well. So lysine has 1, 2, 3, 4, 5. I think that's right. Yes, 5, and then at the very end it has this amino group, which of course can be ionized and can be positively charged. Once again, just like arginine, this has a very, very difficult time becoming unionized. This is like, once again, like between 10 and 12 pKa, this side chain. So not like arginine, there's not like an easy way to remember it. Just lysine, maybe lysine, like lie-scene, like you're a liar. Think of Pinocchio, like Pinocchio's a liar, and like think of like his nose, and his nose gets longer the more he lies. Lysine is a liar. Yeah, I guess that could be a way to remember it. Now, is there a way to remember that lying is positive? I mean, maybe not, but anyway, lysine is one of the positively charged ones, and just like arginine, just like histidine, this one is able to make a salt bridge with glutamate and aspartate. So I'm nearly done. I've got a couple left. So let's see. There's a pretty easy one. So I went over valine. Now leucine. Leucine is a bit of a weird one because leucine has actually two forms in a way. So leucine looks very similar to valine, and I don't think there's like a really easy way to remember it. There's not really, the structure doesn't really make an L shape. I mean, maybe if you really stretch the imagination, this kind of looks like an L, like leucine. Like, I don't know. I think it's a bit of a stretch. But yeah, that's leucine. That's another non-polar amino acid, just like valine. And like I said, just like similar to valine, it has almost the same structure, just one more carbon. And beyond that, so there's leucine, of course, and there's also isoleucine. So isoleucine actually has the unique factor of it is that it has a chiral center. So that's kind of cool. That chiral center does in fact come into play, but depending on the isoform of the amino acid. Anyway, you could also like make the case that like this sort of looks like an L. I don't know. Just to think of like it is, after all, it is, however, an iso group. So isoleucine, it has an iso group, or has this methyl group here at the iso position of this side chain. So okay, and of course, what would be an amino acid? What would be the amino acid groups without glycine? So glycine is the simplest amino acid. It doesn't have a side chain. It just attaches to a hydrogen. So glycine, with that little factoid in mind, this part no longer is it's no longer a chiral center, you know. So glycine has a unique factor. So if like we have like let's say we have a protein. So this is a protein, right? This looks like a protein, I know. So this is supposed to be a protein. So imagine this is an alpha helix. This is another alpha helix. These are a couple of sheets. Glycine has the unique ability to make a turn, right? So imagine right here, right there would be a glycine residue, because it doesn't have the chiral center. It could rotate freely. You're able to make a turn like that. Isn't that kind of cool? In the similar fashion, right? We actually have another kind of unique amino acid where we sacrifice one of the bonding spots on nitrogen. So this would be, excuse me for one moment. This would be NH2 now. It would still be positively charged, but we kind of go around like this. So this would be proline. And the way I remember it is you got to be a pro to attach to the amino group. You got to be a pro to do that. So once again, this has a very unique structural property, such that you were able to make, because this is a far more rigid structure than all the other amino acids, you were able to, like, when you have a proline in a peptide, it is able to make a turn or is able to just have that structural rigidity. It's pretty cool. I think it's pretty neat. Anyway, moving on. We just got one now, I believe. I think we've went through all of them. We're making good time, I think. I think I'm at a half hour around that. Yeah. So, moving on. We got one more, and it's tryptophan. Tryptophan is pretty cool. I like tryptophan. I think it has a cool structure. And also, tryptophan makes tryptamines. And if you know anything about psychedelics, that dimethyltryptamine is very, very potent. And I just think it's so fascinating how, like, you know, we have a structure like tryptophan. Like, I think DMT is able to be secreted at death, which is kind of fascinating. And tryptophan is where that's derived from. So I do think that's pretty cool. Anyway, tryptophan definitely, well, maybe not definitely. You could make the argument that histidine has the most complicated structure. But tryptophan, yeah, it's got a lot going on. This is one of the cyclic. You could say it's a cyclic amino acid because it's similar to phenylalanine, similar to tyrosine. It does have a benzyl group here, and it does have two rings. And that's the thing about tryptophan, is that it's got, you know, the two rings. Which I guess that's a way to memorize what tryptophan is. Well, for one, right? Think about DMT. Think about how hard of a trip you will go on. So tryptophan makes you trip, or you go T for two. It's got two rings, right? So tryptophan, yeah. Anyway, it's got this big old structure, and of course, like I said, this amino acid can be modified similar to histidine. It can be modified into tryptamines, which have their own function in the human body, like I just mentioned. So anyway, I would have some different memorization mnemonics for the lettering. Like if you want to learn the one letter sequences, like alanine A, valine V, methionine M, leucine L, isoleucine I, proline P, and then it gets a little weird. Like threonine, for example, would be T, but of course we also have tryptophan, which starts with the T. Tryptophan is W. Tryptophan is W, and just say it like you're a little kid. So tryptophan, that's that. Yeah, phenylalanine is F. So of course, PH makes the F sound. So phenylalanine. Lysine is K. That's one you just got to remember. Unfortunately, that one just sucks. I guess they ran out of letters for lysine. Arginine, just cut out the A sound. Arginine. Histidine is H. Glutamic acid. Once again, they kind of ran out of letters for these. But glutamic acid is also glutamate, right? You can't really see my handwriting. Glutamate, which ends with an E, so that's it. It's E. Glutamic acid, glutamate. That's E. And aspartic acid, just try to enunciate the T in an aspartic more. So aspartic acid. Aspartic acid is D. And asparagine and glutamine, just think glutamine, glutamine, so that's Q. And asparagine, honestly, this one is I'm blanking on asparagine if I'm being honest. I gotta look that up real quick. Sorry, asparagine is N. So asparagine is N. See, I feel like I have a good grasp of my amino acids, and even I have trouble sometimes because they're so complicated. Anyway, threonine, that one's simple. It's just T. Cysteine, again, simple, C. Serine, S. Tyrosine, since we already have threonine, tyrosine, we just take the Y. Tyrosine. So that's a Y. Glycine is G. And proline, and like I said, proline, methionine, leucine, isoleucine, valine, and alanine. So that's your amino acid review. Hopefully this helped you. Feel free to share. In fact, I would really like it if you did share, and I hope this helped. Bye.